Confusing the Golgi with the ER, or forgetting what the lysosome actually does, can cost you easy points on the MCAT. But what if you could see cell organelles as a connected system instead of random facts to memorize? In this quick, high-yield guide, we'll break down each major organelle, its structure, its function, and exactly how it shows up in MCAT questions so you can recognize patterns, not just definitions. Let's turn cell biology into something that actually sticks. The nucleus houses genetic material and controls gene expression through its double membrane structure and nuclear pores. The nucleus is often described as the control center of the cell because it contains DNA organized into chromatin, which can condense into chromosomes during cell division. Structurally, it is surrounded by a nuclear envelope made of two lipid bilayers, creating a barrier that separates transcription from translation, a key concept in eukaryotic cells. Embedded within this envelope are nuclear pores, which regulate the movement of molecules like mRNA and ribosomal subunits between the nucleus and cytoplasm. Inside, the nucleolus is responsible for producing ribosomal RNA, making it essential for ribosome assembly. On the MCAT, you are often asked to connect structure to function, such as why transcription occurs in the nucleus while translation occurs in the cytoplasm. This spatial separation allows for regulation and processing of RNA, including splicing. A helpful mnemonic is DNA stays, RNA strays, reminding you that DNA remains in the nucleus while RNA exits. This topic also connects to gene regulation, cell cycle control, and diseases like cancer, where nuclear processes are disrupted. The mitochondria generate ATP through oxidative phosphorylation and contain their own DNA, reflecting their evolutionary origin. Mitochondria are known as the powerhouse of the cell because they convert energy from nutrients into ATP, the cell's usable energy currency. They have a unique double membrane structure. The outer membrane is smooth, while the inner membrane is folded into cristae, increasing surface area for the electron transport chain. Inside is the mitochondrial matrix, where the Krebs cycle occurs. The presence of circular DNA and their own ribosomes supports the endosymbiotic theory, which suggests mitochondria evolved from free-living prokaryotes. On the MCAT, questions often focus on linking structure to energy production, such as how increased cristae surface area boosts ATP yield. You may also see experimental setups involving oxygen consumption or metabolic poisons that disrupt the electron transport chain. A helpful way to remember is mito equals mighty energy. This concept ties closely to metabolism, cellular respiration, and apoptosis, as mitochondria also play a role in programmed cell death. The endoplasmic reticulum is divided into rough and smooth regions that specialize in protein synthesis and lipid metabolism, respectively. The rough endoplasmic reticulum, or rough ER, is studded with ribosomes on its surface, giving it a bumpy appearance and making it the primary site for synthesis of proteins destined for secretion, membranes, or lysosomes. In contrast, the smooth endoplasmic reticulum lacks ribosomes and is involved in lipid synthesis, phospholipid production, detoxification, and calcium storage, especially in muscle cells where it's called the sarcoplasmic reticulum. The MCAT often tests your understanding of how proteins move through the endomembrane system, starting in the rough ER, then to the Golgi apparatus, and finally to their destination. A classic question might involve, what happens if the rough ER is damaged? Protein secretion would be impaired. A useful mnemonic is rough equals ribosomes, smooth equals steroids. This topic connects to membrane biology, protein trafficking, and liver detoxification processes, all of which are high yield for the exam. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport to their correct cellular destinations. Structurally, The Golgi is composed of stacked membrane-bound sacs called cisternae with a distinct polarity. The cis face receives materials from the ER, and the trans face ships them out to various locations. As proteins pass through, they undergo post-translational modifications such as glycosylation, which can affect their function and targeting. One critical function is tagging proteins with mannose, 6-phosphate, directing them to lysosomes. On the MCAT, you might encounter questions about protein trafficking errors, such as what happens when lysosomal enzymes are not properly tagged, leading to diseases like eye cell disease. A helpful way to think about the Golgi is as a post office that labels and sends packages. This organelle is central to the endomembrane system and connects directly to secretion, membrane renewal, and intracellular transport. Lysosomes and peroxisomes are specialized organelles responsible for intracellular digestion and detoxification processes. Lysosomes contain hydrolytic enzymes that function optimally at acidic pH, allowing them to break down macromolecules, damaged organelles, and pathogens in a process known as autophagy. Their acidic environment is maintained by proton pumps in the membrane. Peroxisomes, on the other hand, are involved in oxidation reactions, including the breakdown of very long-chain fatty acids and detoxification through hydrogen peroxide metabolism. They contain enzymes like catalase to neutralize reactive oxygen species. On the MCAT, you may be asked to distinguish between these organelles or predict the effects of enzyme deficiencies, such as lysosomal storage diseases. A useful mnemonic is lysosome equals lysis, peroxisome equals peroxide. These organelles connect to metabolism, oxidative stress, and cellular homeostasis, making them important for understanding disease mechanisms and biochemical pathways.